Please, I did start your iClicker. So here, let's go and see what we're going to cover today on the last part here of your immune system. So we stopped at this slide before when we were talking about antigens, right? Does everybody remember this from previous life before spring break? Hopefully, yeah. So in my case, it was basically like before the exam, right? Because I wasn't here for the exam. So, okay. Oh yeah, and also, all right. So we're talking about antigens, and we're going to talk about antibodies, and then about humoral and cell-mediated immune responses, right? So here, when you're talking about antigens, we are basically talking about something that can cause reaction of your immune system, in particular adaptive immune system reaction. Okay, so you have an antigen that is a whole blob of stuff. So each antigen can have more than one epitopes. So as you see here, epitopes are also called antigenic determinants. Okay, so this antigen here, as you see, it has 3 different epitopes. So epitopes is a little piece of this big antigen that can be recognized by, in this case, by an antibody. Okay, so epitope is a piece of your big antigen. All right, so what is important to know here? Okay. All right. So a molecule that is called the antigen, it is basically going to cause this adaptive response of your immune system, and is going to lead to a memory of your immune system towards that particular antigen. Okay. So when you say a molecule is capable of causing immune response memory, you're basically talking about mainly about proteins. Okay, there are other antigens that are made of lipids or carbohydrates, but you're mainly talking about proteins. Now, there are some certain types of compounds or certain chemicals that do not get recognized by antibodies or by other cells of adaptive response, mainly by T cells. And those are really big polymers. Okay, so when you look at plastics, for example, there's like this huge polymer. Those do not get recognized by antibodies or by T cells. That doesn't mean that those molecules do not get recognized at all. They do get recognized, and you do have a certain form of immune reaction that is built around them, but those are based on innate immune cell reaction. Okay, so it's not like you put a plastic in your body and nothing will ever recognize it. No, you have your innate immune cells that go there and recognize that that thing probably wasn't supposed to be there, and those innate immune cells start to build to deposit some fibers and build a kind of capsule around that strange object that is in there. Okay, so when you talk about really antigenic determinants or antigens, you're really talking about the function of your adaptive immune system, not really of your innate immune system. All right. So here, looking at T cells and B cells, okay, each of those cells have receptors on their surface, and each of the receptors, let's say if you have more than one T cell here, hold your T cell here with a receptor here, each one of the receptors on a particular T cell is made to recognize only one antigen. Okay, so this T cell here is going to recognize an antigen, let's say that it is like blue. And then this other T cell here recognizes only this little antigen right there. Okay, So every T cell can recognize only one little epitope on an antigen, okay? And the same goes for the B cells. So B cells have the B cell receptor. Each B cell has a unique B cell receptor, the same way that each T cell has a unique T cell receptor. So each B cell is going to recognize only one epitope. Okay, so get this idea in your mind, okay? Because the situation is the following. You have many different B cells. Okay, so we do here B cells, a lot of B cells. You have a ton of B cells. Each one of these B cells have one very unique receptor on the surface, and each B cell is going to have only one type of receptor that recognizes only one type of epitope. Okay, so the receptors in this B cell, let's say that this guy recognizes a purple epitope. So each B cell receptor is only going to recognize this little purple epitope. Okay? If you look at another B cell here, this B cell is going to have another unique antigen recognition receptor. Okay? So all the receptors on this B cell is going to recognize only one epitope. So this guy is going to recognize something different. He's going to recognize a green epitope. Okay? So every one of those receptors can only recognize that little green epitope. It cannot recognize anything else. So each B cell is very specific for only one epitope. Same thing goes with the T cells. Each T cell has receptors that are going to recognize only one epitope. Okay? So then what's going to happen here? When you have your B cells that, let's say, the B cell can phagocytose, so it ate a virus that was out on the floating in the blood. It digested the virus, it cut little pieces, it put a piece of a protein of the virus on its presenting molecule that is called MHC class 2. Okay, and it presented a piece of that protein to a T cell. A lot of T cells that do not recognize the protein are going to be around, so nothing is going to happen. But if the one T cell that is meant to recognize that little piece of protein comes along and binds to it, okay, on the antigen, on the MHC molecule, then you're going to have matching of recognition of that epitope, okay? And this is when you're going to have mutual activation of those cells. Okay, this is when you're going to tell your T cell to start to become an acting cell or an effector cell. Okay, and then the T cell is also going to tell the B cell to do something to help to destroy that pathogen. Okay, so each one of your T cells and each one of your B cells is going to recognize, again, only one epitope. All right. So here you say, okay, what are the types of cells that can recognize foreign stuff, right? What is it that, how is it that you're going to recognize antigens or epitopes? So you basically have these two types of cells that are lymphocytes. So those are your B cells and your T cells, okay? And you have these other types of cells that are your antigen presenting cells. Okay, most of your antigen presenting cells that are most important are really part of your innate system. So those are your dendritic cells, okay? But then you also have some other antigen presenting cells that are like the B cells, or even some endothelial cells can also be can be presenting antigen. Okay, so let's see here what is the main idea of these things here. Okay, So let's first talk here about your lymphocytes, right? So when you talk about lymphocytes, this, as you remember from the blood chapter, right? Those come from the lymphoid lineage of cell development, and they start to develop in the bone marrow. So all lymphocytes start to develop in the bone marrow, but then some lymphocytes are not going to finish the development in the bone marrow. The ones that finish the development in the bone marrow are only the B cells. The T cells are going to come out of the bone marrow and they migrate to the thymus and they finish their development there. So the T cells are going to go to this other primary lymphoid tissue that is your thymus, and they're going to go through that thymic school. They're going to learn how to recognize MHC molecules, and they're going to learn how not to attack your own proteins. Okay? So why is this important? Why do you have to teach your lymphocytes to only attack foreign things, right? First reason is that they have to be able to cause an immune response. So they have to be able to recognize something, okay? What is they going to recognize? Well, each one of those cells is going to recognize only one epitope. So they have to recognize the epitope that is foreign, right? They don't want, your body doesn't want your lymphocytes to recognize and attack your own tissues, right? So this is first reason, so that's called immunocompetence. Another reason that you have to teach those lymphocytes how to behave is that they cannot be attacking you. So if you have some lymphocytes that during development is found that they attack self, they're going to get killed. Okay, so this is the self-tolerance part of teaching the lymphocytes how to behave. So what's the idea here? First thing, attack foreign stuff. Second thing, do not attack self. Right? Okay. So let's go here through the development and maturation. So remember, both of them are developing in the red bone marrow. B cells continue to mature in the red bone marrow, and they still have a further maturation process that happens mainly at the spleen or at lymph nodes, but most of the development of your B cell is going to be in the bone marrow. Okay, and then your T cell, it goes to the thymus and develops in the thymus, and once it is checked that it's not harmful, it's going to come out of the thymus and it's going to go into circulation, it's going to go through the lymph, get to the lymph nodes, and then it's going to go back to the circulation and it's going to keep on looking for the antigen that is meant to recognize. So T cells like to travel a lot. B cells like to be sitting at the secondary lymphoid tissues. They also travel a little bit, but not too much. Right? Remember that story that if you have both of them traveling in the blood, they'll never meet each other? Right? So yeah, you're running with the B cell and the other cell trying to catch up with the other B cells and never meet. So this is the idea why T cells travel a lot, but then the B cells like to be sitting in the secondary lymphoid tissue. So let's see what happens here at the secondary lymphoid tissue when your lymphocytes recognize their proper epitope. Remember, it's going to be only one lymphocyte that's going to recognize that epitope. And then you're thinking, this epitope came from an antigen that is part of a pathogen. How many of those epitopes do you have around in your blood? This is a reasoning question. Okay, it was actually a question. Did everybody hear the question? Okay, answer. I'm not sure if I brought chocolate. Ask the question again. Okay. The question was, you have only one B cell and only one T cell that can recognize an epitope. Where did the epitope come from? A pathogen. How many of those epitopes do you think you have in your body? A lot. Why?
They replicate, and the virus or bacteria will spread.
Exactly. Everybody got that? Yeah? Because if you're able to recognize a little, oh, you gotta, hopefully you like this candy. You have a pathogen in your body. If that pathogen got to a secondary lymphoid tissue, it means that first of all it's traveling, and first of all it got into your system, and second it's traveling, and third there are many more of those around. Now you have only one cell of a B kind that can recognize it. You have only one cell of the T kind that can recognize it. And what are they going to do if you have a lot of pathogens and only one B cell that can recognize it? You had a question.
I'll let you finish because I'm not sure yet.
Oh, okay. So now, only one B cell, only one T cell, and lots of pathogens. Can you win that battle? No. So each one of those cells of one B cell and one T cell that recognize your pathogen, they're going to have to multiply a lot and fast. Okay? So this is what you call clonal selection and also clonal expansion. Okay? It's not an M, that is an N. Okay. Clonal expansion. That thing is going to divide very quickly because you need a whole army of those B cells and T cells that will be able to fight that pathogen. Okay? So this is your clonal expansion or clonal selection. So one cell recognized, it keeps on dividing, and after they recognize the epitope, after they recognize what they're supposed to recognize, they are going to become effector cells. Because before they recognize the pathogen, before they recognize what they're supposed to, they are just naive. They have never seen a pathogen before. They're totally naive. They don't do anything. They just hang around. After they get activated by recognition of their epitope, they divide and they become effector cells. So your B cells are going to become plasma cells, and they are also going to become memory cells. You're going to have B memory cells. The plasma cells are going to shoot out antibodies. The memory cells are going to hang around for a very long time, most likely for your whole life. They hang around mainly like back in the bone marrow, or they stay in the secondary lymphoid tissues, but they're going to be there just waiting in case that pathogen shows up again some other time. Okay? So after activation, you're going to have effector cells, and you're also going to produce memory cells. The same thing happens with your T cells. After they recognize that epitope, they are going to become effector cells. There are many types of effector T cells around, okay? And they're also going to become memory T cells, okay? Everybody cool with that? Okay. Now, so here, how is it that your T cells are going to learn not to attack you, and how is it that they are going to bring up a secondary immune response? Okay, so I briefly had mentioned this before when we talked a little bit about different cell types, the blood cell types, right? So here, when we talk about in specific T cell development, right? We know that they're developing in the thymus, right? And T cells, they are a bit more. They have to go through more schooling than the B cells do, okay? Because you got to remember that T cells, they see the antigen differently when you compare to how B cells see the antigens, okay? So a little bit before we get to the T cell thing, when you look at the B cell development, you know everything is in the bone marrow, so the B cells are going to see the antigen basically in the native shape of the antigen. So they're going to see in 3D, okay? B cell receptors, they are able to interact with the antigen and see the whole blob of little protein that is the epitope of that antigen, okay? T cells, they don't do that. They are going to see the antigen in 2D. They're going to see a little line, a little stretch of a polypeptide that is a certain size and is stretched out. T cells cannot really see the antigens in 3D. I mean, the T cells that are circulating in your blood, the alpha beta T cells that we're used to. Okay, so T cells have to be antigen that is stretched out, right? B cells, no, they see 3D. So when you talk about B cell development, developing the bone marrow, inside the bone marrow, you're going to have supporting cells that are going to teach your B cells what is self and what B cell should not attack. Okay? So in the bone marrow, you have these antigen presenting cells that are going to show to the B cell this natural shape of the antigen. So we're talking about what are these? Dendritic cells. Okay, they're going to show it to the B cell. This is what this thing looks like. Do not attack it because this is your self stuff. Okay, so here you're going to have inside of your bone marrow things that are in 3D. All right, this is in 3D. Okay, so B cell has the receptor. Remember, receptor in the B cell is an antibody. That's your IgM antibody, also IgD. So you have your antibody with two arms. So antigen is going to get there. This is self-antigen. It's going to get to the B cell receptor. B cell receptor is not supposed to be catching it. If it catches it, and this is self-antigen, what's going to happen to the B cell? It's going to die because it's not supposed to detect self. Okay, if it detects self, B cell is going to die. So that is your negative selection of B cells in the bone marrow. Okay? I don't have it in a slide for that, but you gotta know that. Okay, I'm telling you now. B cells undergo negative selection. And I remember I didn't put a slide because it was a simpler thing to remember. Okay. So now we go to the T cell development. T cell starts developing the bone marrow, and then they go to the thymus. When they get to the thymus, they got a lot of work to do. They have a lot of schooling to go through. Okay, so first thing that the T cells are going to learn in the thymus is to recognize presentation by MHC molecule. Because remember, T cell has to see things in 2D. So this is a line. I'll stretch the peptide. So this is not 3D, it's only 2D. Okay. When the T cell gets there, it has to recognize the MHC, right? Either MHC1 or MHC2, but it has to interact to the MHC. If it does not interact to the MHC, the T cell is useless. So what's going to happen to the T cell? It's going to die. Okay. If the T cell gets there and recognizes the MHC, okay, so this is my MHC, and this is my antigen presentation molecule, either one or two, okay? Doesn't have anything on its groove, it's not presenting anything, it's only the MHC, okay? So here's MHC. Now, when the T cell gets there, if it doesn't recognize it, it's useless, it's gonna die. If it gets there and it really recognizes it, and it doesn't want to let go, but there is no antigen, that is a problem. Okay? It is a problem because that T cell is so willing to grab this MHC that if it comes out of the thymus, it's going to see MHC on your cells and it's going to bind to it and it's going to get activated. That's a big problem. Okay? That T cell is dangerous. So what you got to do with it? Kill it in the thymus. Don't let it out. Okay? All right. So now, let's say that the ones that didn't recognize get to die. The ones that bind too strongly, they get to die. Now you're going to have the ones that interact just right, and those are good. Those are going to learn some more stuff. They survived the first stage. So the first stage of recognizing your MHC just right, that is called positive selection. Okay, everybody got that? Okay, am I going to ask you about that? Yes, okay. All right, so now comes the second stage of the development. So let's put it here. Now this thing is going to present self molecule. Okay? So now you got a T cell that is so happy, it just survived the first stage of death over there. It goes in here and say, oh, it is a self molecule. Do I want to interact with it? No. I know what's going to happen. So that guy survives. Now comes this other guy. I passed the first stage and look, ooh, it's an antigen. I'm going to interact with it. What is going to happen with this guy? There's only one answer to that question, right? It's going to die. You're not supposed to recognize your self-antigen. You recognize your self-antigen, you get killed. So that is called negative selection, okay, for your T cells. So T cells undergo positive selection and negative selection. B cells only undergo negative selection, okay? So this is what is written in this slide. Okay. Oh, there's something else. Your T cells can only interact with your own MHCs. Okay. If you get a T cell from somebody else, it is useless for your body because that T cell is not going to interact appropriately with your own MHC. So you have unique T cell receptor, you also have unique MHCs, and you got a bunch of them. We're not even going to start talking about those guys, okay? You have a lot of different MHCs, like 6 different types that are expressed in every one of your cells, or actually 3 for MHC1. It gets complicated. If you're curious about that, take immunology. But you have a lot of different MHC molecules. So chances of you finding somebody else who has exactly the same MHC molecules, those are very low chances. This is why it's so difficult to find a compatible match in case of organ transplant. It's because of those MHC molecules. Okay? If you get an organ from somebody who does not have matching MHC, your cells are going to attack that organ because it's going to recognize the MHC as being foreign stuff and is trained to attack foreign stuff. Okay, so that's a problem. Okay, can you get 100% match of organs in case for transplantation? Can you get total 100% match? You can, but it's extremely difficult. Let's say that by a lot of luck, you get all of the MHCs to match. You still have those minor histocompatibility complexes that we're not even talking about. Okay, it's very difficult. So there are always some lymphocytes that will be recognizing some organ that comes from somebody else. And what you do in order not to kill the organ is you take immunosuppressant drugs. Okay, so everyone who has an organ transplant needs to take immunosuppressant drugs. Okay, so your body will not attack the organ. Okay. So uniqueness of your MHCs, uniqueness of your T cell receptor that recognizes that. So I did mention a lot of stuff in this slide. Okay, make sure you understand most stuff that I mentioned here. All right. So here, then we get to these antigen presenting cells. The most important one is your dendritic cell. Okay, dendritic cell, that's your most important guy. Because those are made to activate your naive T cells, the ones that have never seen antigen before. Okay, and they're very good at doing that. So dendritic cells are very good at activating your naive T cells because dendritic cells are going to phagocytose some pathogen. It's going to break it up, right? Kill it, chop into little pieces, put it on MHC class 2 and present it to your T cell. And it has other things on its surface that is going to interact with the T cell to activate it, naive T cell. Then the naive T cell is going to become an effector T cell. Okay? So the dendritic cell presents to naive T cell. Naive T cell is going to be experienced is going to become effector T cell. Okay. Besides dendritic cells, you have other cells that have MHC class II on their surface. So the whole idea of this game is antigen presenting cells have to have MHC class II on their surface. Okay. You have other cells that also have MHC class II on their surface, and one of them is here, your macrophage. Okay. And you also have B cells that have MHC class II on their surface. They naturally have it. Okay, they don't need a push to express those. They already express them. Besides those cells, you also have other types of cells in your body, especially cells that are lining of your blood vessels. Okay, so endothelial cells. Those normally do not present MHC class II. But if you have inflammation going on, then those cells can start expressing MHC class II. Okay, so you have other types of cells that eventually, because of infection inflammation, they can start presenting MHC class II. But they are not professional antigen presenting cells. When you talk about professional or antigen presenting cells, the real professional one is really your dendritic cell. Then the other ones are, macrophages and B cells. Okay? Those are really, really important for presenting antigen to T cells. All right. So what's going to happen here? What after you have recognized the antigen, right? So here, let's see, here you have your dendritic cells. Usually dendritic cells are depicted like that. We like lots of tentacles, right? So here you have your dendritic cell that is going to present the antigen on MHC class II. Is going to present it to your T cell. T cell is going to get activated, okay? If the T cell is a cytotoxic T cell, meaning that it has CD8 on the surface, if you have a cytotoxic T cell, actually you're going to see the antigen presented on MHC class I, okay? And this T cell is going to become an effector cell, and that means it's going to be able to do the job that it's supposed to do, which is kill stuff. Okay, so cytotoxic T cell kills infected cells, but it's not going to start killing infected cells unless it is activated by a dendritic cell. Okay. Now let's say this dendritic cell brought a little antigen, brought actually the whole pathogen here to your B cell and presented the pathogen to the B cell. Okay. The dendritic cell brings the whole pathogen. It doesn't chew it up to present it to the B cell. Okay. It brought the whole thing, gave it to the B cell. The B cell ate it up, chopped it up, phagocytosed, and get activated by this dendritic cell. Okay, the B cell is also going to become effector cell. So the B cell is going to be transformed into a plasma cell. That's your effector cell of B cell, and also memory cells. And then the B cell is going to start to shoot out antibodies to bind to the epitope that is part of a pathogen. Okay, so what is happening here? When your B cell gets activated by the pathogen that was brought by your dendritic cell, then your B cell is going to shoot out the antibodies, and this is what you call humoral response of your adaptive system. So this whole side here with antibodies is humoral response. Okay? Now when your dendritic cell brings your pathogen and presents to the T cell, in this case here it was a MHC1 to CD8, but it also presents on MHC2 to CD4 T cells, right, naive T cells. Now those T cells are going to get activated, and this branch here is what you call the cell mediated branch of your adaptive immune system. Okay, so when you're talking about B cells producing antibodies, that is humoral response. When you talk about activating T cells, that is your cell mediated. So what that means, if it is cell mediated, that means the cell itself is going to interact with a molecule present on another cell and will be able to directly, by punching the other guy, is going to kill it. Now, humoral response means your B cells produce antibodies that bind to your virus or bacteria or pathogen, but the B cell itself never interacts with something that is infected. Okay, so the B cells shoot out antibodies and it sends these little workers to do their jobs. Okay, everybody got that? Cell mediated, humoral, okay, two different things. Okay. Oh, here, finally, your first question. Let's get your eye clickers going. When this thing works, hopefully today. Come on. Eye clicker is not responding. Okay, wait for the program to respond. So in the meantime, let's answer the question until the program wakes up. Okay. The process of attaching antibodies or complement proteins, okay, to the bacterial cell wall to enhance phagocytosis is called what? And now the thing disappeared. Okay, let me see here. Select. It seems that it's working. Okay. What is your answer? D. That is your opsonization. Opsonization. No. So here, talking a bit more about your cellular immune response. That's the job of your T cells. Okay. So your T cells are going to be able to detect when a certain cell has an intracellular pathogen. Intracellular pathogen. So it can be virus or it can be intracellular bacteria. Okay. So T cells are going to detect the presence of something strange that is inside of the cell. Like how? So the T cell is going to see on the MHC molecule if it is here. Let me see here. This guy here is, let's say MHC1. Okay. So your T cell is going to go in there, is going to detect this presence on MHC1, and is going to say, oh, you have something inside of you that shouldn't be there. So I'm going to kill you. Okay, that's what your cytotoxic T cell does. Detect something on MHC1 that is not supposed to be there, and it kills the cell that presented it. Okay? Keep in mind that when you have those MHC1 on the surface of cells, they always have something. They always have a little peptide stuck to them. You're never going to find empty MHC1. And which ones are the cells that have MHC1? It presents self, but which cells in your body have MHC1 molecule? I got a Copico candy, cappuccino, a little bit of caffeine. Take a guess. Yeah. These cells have MHC1, but they're going to recognize MHC1, but that's not the answer. But you get a candy. Okay, let's think about it. Big one. Okay, so let's go back to beginning of the class and put the question in a different way. Which are the cells that do not have MHC1? You should know that like right away. Yeah, but that's naturally in your body. They're not infected and they don't have MHC1. You know. put her on the spot. I don't like to do that. Okay, so if somebody does that to my kids, then and I get mad about that. Okay. Who knows? Yeah. No.
Would it be an ectomegalyte?
No. Yes! Did everybody hear that? Not they are screaming, but the type of cell. Okay, everybody wants? Red blood cells. They do not have MHC1. They also don't have a nucleus. Yeah. Yes, all nucleated cells have MHC1, right? Everybody got that? If your cell has a nucleus, it has MHC1, unless it's sick or infected, okay? MHC1 is never going to be empty. It's always going to present something, self. If T cell cytotoxic gets there and recognizes something, it's going to kill that cell that presented something strange, okay? Could it be that the MHC1 is presenting a self molecule on its surface? Yes. All of your cells have MHC1. MHC1s are never empty. All of your cells are going to present self-antigen all the time. All the time. Okay? So why is it that your T cells don't go around killing your own cells? That's a fundamental question. Everybody got to get this idea right in the head. Anyone?
I'm sorry, can you say it again?
The question, if all of your nucleated cells have MHC1, MHC1 are never empty, they're always presenting self-antigen. How is it that T cells don't go there and interact and kill those cells? Yeah, that's what the idea I was looking for. You are not going to have in your body, in your circulation, you are not going to have a T cell that can do that, because all of the T cells that could recognize self got killed in your thymus by negative selection. This is why your immune system doesn't kill you. That was a good reason, right? So if you have a cell that does not get killed in the thymus, and that cell can recognize self, and it comes out of the thymus, you're in trouble. Okay? Because that cell is eventually going to kill the target tissue or organ, and you're going to have an autoimmune disease, and that's trouble. Okay? All right. So those are your T cells. T cells are going to look for intracellular pathogens, and they're also able to detect cancer cells, because cancer cells, they start to produce proteins that are not normal. So they were not there when you were born, they got transformed, right? If they present something that is strange, then your T cell should be able to recognize it, because something that is strange wasn't presented in your thymus, okay? So if the T cell cytotoxic sees something strange being presented, it should go and kill the cell, because it's thinking that is either cancerous or is infected, okay? All right, so here, major type of your T cells. You have your helper cells. They have CD4. You have your cytotoxic cells. Those have CD8. CD8 cells are the ones that when they recognize something, they go and they kill the cell. So they're called cytotoxic because they have toxic stuff to kill cells, okay? Helper cells do not do the killing most of the time. Helper cells, they're going to help other cells to get activated. They're going to dictate how your immune reaction is going to take place. Okay, so you have the helpers and you have the killers, different types of your T cells. Besides that, you also have formation of your memory cells. So you're going to have effector cells and you're going to have memory T cells. They're going to hang around like forever, hopefully. Okay. Right, so here's a little overview of the different types of immune reactions and the different types of cells. So when you're talking about B cells, you know you're going to have humoral immune reaction, and they are going to secrete antibodies in comparison to your T cells that do not secrete antibodies. So the receptors on the T cells, they never come out of the cell membrane. They stay always stuck on the cell membrane. But then the T cells, they do the cell-to-cell contact and combat, so you call that cellular immunity. Major difference also is that T cells are going to fight intracellular pathogens, while your B cells are going to find stuff that is outside of the cells. All right. Another important difference, your T cells are going to finish the development in the thymus, contrast to the bone marrow. Okay. And effector cells. Your T cells are going to have your cytotoxic T cells and your helper T cells. I forgot to talk about the regulatory T cells, but yeah, so, you know, regulatory, what does these guys do in here? These guys are a bit different. They only come up at the end of a fight with a pathogen. They come up because you don't want this immune response to be happening forever in your body. You want to get the effector cells, get them to do the job, eliminate the pathogen, and disappear. Okay? Because you don't want these highly reactive cells to be in your body all the time, otherwise you're going to develop chronic inflammation, and that's not good. Okay? So the cells that did the job, they will have to die, or they will have to become zombies. Meaning that they'll hang around for some time, like semi-dead. You know what a zombie is, right? Hopefully, okay. Zombie, the guy's like, you know, I'm almost dead. I'm not dead yet, but I'm not doing much. I never seen zombies doing anything except in movies, right? But yeah, zombies are not supposed to do much. They could scare other cells around, but they're not going to do much. Eventually, they will die, really die. Okay. Regulatory T cells will get those highly active cells to become zombies or to die. Okay, so regulatory T cells want everybody quiet. They want to quiet down that immune reaction. Okay, so just reiterate here, types of T cells: cytotoxic, kill the cell; helper T cells, these are doing the major battle. When the battle is over, the regulatory T cells will come along and say, all right, guys, quiet down. There's no more business here. There's no more pathogen. Just, you know, zombie around for some time and then just, you know, die. Right? So that's the job of your regulatory T cells. Yeah. They will quiet down the fighting as long as you don't have more pathogens hanging around. Okay? Okay, and so those are your effector T cells. And then here for effector B cells, you have plasma cells. Okay? Besides that, you also have your memory cells. So you have B memory cells and you have T memory cells. All right. So here, role of those MHC molecules, right? So these are the things that are also important to know about this type of responses, right? Class 1 MHC, remember, is present in all nucleated cells. Class 2 is going to be present in your antigen-presenting cells. Okay? Class 1 is going to interact with CD8 T cells. Class 2 with CD4 T cells. And you know the rule, right? You gotta get multiplication of those interactions, and you're gonna get 8. Did I mention this here before? Class 1 times CD4. No, class 1 times CD8. You see? Everybody makes a mistake. Equals to 8. Class 2 times CD4 also equals to 8. So your multiplication result should be 8 for those interactions of CDs and MHCs. Okay, in case I didn't mention it here, so now you have it. All right. Okay, so class 1 MHC is going to be concerned with stuff that is inside of the cell. Class 2 will be concerned with pathogens that are outside of the cell. And you can read through this and make sure that you understand what they're doing. Okay, another question. Okay. The adaptive immune system involves 3 major cell types. So you have here your antigen-presenting cells, T cells that are going to constitute which part of your immune system response, and then B cells are going to be what part of immune system response. Okay, what is your answer? D. Okay, some people answered A. Now, when you are talking about T cells and B cells, everything is specific because each one of them is going to have a very specific receptor on their surface. Okay, everything is specific here. All right. So here is a slide showing you the specificity for clonal selection, right? And let me see where I put this here. All right, this was just to emphasize that you have only one type of B cell that is capable of recognizing one epitope on an antigen. So when this cell recognizes the epitope, it gets activated and it divides a lot. That's your clonal selection, and is going to produce one type of antibody. Okay? Because it's one type of cell is producing only one type of antibody at this stage. Yes.
So in the entire body, there's one B cell per epitope for everyone, or are we just talking about like a localized area?
No, practically speaking, you're gonna have one, but you have several epitopes in each of antigen. So yeah, I mean there are so many different types of B cells and T cells around that even if you don't have one that recognizes a particular epitope, it's going to have an epitope that recognizes that antigen from some other side.
Okay,
Yeah.
I feel like that would be risky if it was just one.
Yeah, it's one, maybe the most like 10 cells, and it's still risky, but this is why clonal selection is so important. Because when it gets activated, they have to divide quickly. Like not as quickly as we would like at first time, but they divide a lot. Okay? Yeah, it's kind of puzzling, right? Like, imagine if you have only one cell that can recognize one virus, like the whole virus. That is risky. No, but the virus will have a lot of epitopes, so eventually you're going to have one cell that's going to recognize something. Okay. All right. Okay. So here is still on the clonal selection B cell. All right. All right. So now we get to those primary and secondary responses. Right. So at first, when your pathogen crossed the skin or the mucosal membrane and it got into your system, so it got over or it surpassed all of those innate immune defenses. So now you are going to get to presentation in the secondary lymph organs. You're going to start activating your B cells and your T cells, right? But it takes time for those cells to, first for them to recognize the pathogen. Then they're going to have to get activated. Then they're going to have to start dividing. They're going to become effector cells. They're going to make some memory cells in the meantime. Effector cells have to start doing their effect. They have to travel from the secondary lymphoid tissue all the way to the site of the infection. They're going to have to start killing the pathogen. In the meantime, if it is the first time you're seeing the pathogen, you're going to have to wait for all this stuff to happen. And how long does it take? About 10 days. Okay, 10 days for you to have a very good adaptive immune response to that pathogen. In the meantime, your innate immune system is working, your complement system is working, you're trying to get rid of the pathogen somehow. But then after about 10 days, when your adaptive immune system picks up, it will take over and eliminate the pathogen. So you start thinking, anybody here had the cold before? Like a cold, yeah, or flu? Flu? Do you feel miserable with the flu? Yeah, your body hurts, and sometimes you have a fever, and it doesn't feel good. And how long do you feel like that? How long? I mean, how long does the flu take to go away? About a week, more or less like, oh, look at that, 10 days. Oh, why? Why it takes 10 days? Because at that time, your adaptive immune system will come into action and eliminate the virus, right? 10 days, roughly, give or take 10 days. Now, let's say that next flu season, if you would be lucky enough to have the same virus that showed up a year ago to show up now, you would not get sick because you have those memory cells, okay? So if you get the virus again in your system, the one that you got last year, exactly the same virus without any mutation, right? You just forget about it. You never even notice that you got the virus because your immune system will jump into action, eliminate the virus like within a day or two. Okay, you don't get sick. Okay? But the first time that you were exposed to the virus, that is called priming. Okay, because you've never seen it before, that is called priming, and you're going to have your primary immune response happening. And that's the one that takes 10 days. Okay? By the time you get to seeing the virus again, that's when you're going to have your secondary immune response that happens quite quickly. So that thing is faster, okay, is more efficient, but it's not going to happen if you have not seen the virus before, okay? How is it possible that you have this secondary immune response? Well, it is possible because you have those memory cells that hang around forever, okay? They don't die. They just, I mean, they can be replaced by the same type of cells, so they stay around for a very, very long time. Okay. This secondary response takes 2 to 3 days or so. So here looking at the graph, okay, what does this mean? This means that at first you are exposed to the virus, your body has never seen it. At first your innate system is going to try to get rid of it. If it is not successful getting rid of it, and the immune system is going to present the virus, right, to your lymphoid cells, right, to your lymphocytes. So here, after 7 to 10 days of activation here of your lymphocytes, that's when you're going to have your antibody response, antibody production, primary immune response, and then you're going to get rid of the virus, and everybody's going to be quiet down because at this point your regulatory T cells are going to tell everybody to just quiet down and relax, okay? Now let's say that after you have recovered from the first infection, you have seen the virus again, the same virus. Now, look at this, quickly you're going to have this secondary immune response. Very quickly, because your memory cells are ready to fire, okay? But now let's make it a little more complicated. At the same time, like on the same day that you have seen this first virus, now you're also going to be exposed to another cold virus, okay? You got some mutations, but this second virus cannot be recognized by memory cells because it's new to your system. It has new proteins, the virus mutated, okay? So now you're going to have to start to develop the cells. You're going to have to get effector cells that recognize that specific second virus. And it's the first time you're seeing virus B. You have seen virus A before, now you're seeing virus A and B. For virus A, you're going to have a very fast secondary response. For virus B, the whole thing is going to start again. You're going to take 7 to 10 days to get enough antibodies to get your primary response to virus B. Okay? So now let's say that after 28 days, okay, so here are your T cells, regulatory T cells, they're coming along and quieting everybody down. Regulatory T cells are working on that too, and it's going to take a little more time to get that graph down. Let's say here, if you get exposed to virus B again, what is going to happen? Quick response. You're going to have your secondary response to virus B. Okay? And this is what is a definition here of your immunological memory. Okay. Another thing that happens here with a second exposure to the same virus, not only you have a faster response, but your antibodies get better. Okay, you're going to have antibodies that are going to be able to grab that antigen in a better way than you had with the first antibodies that were produced over here. Okay, so you have fast response and you have better antibodies that are going to recognize what it has seen already before, what it was already primed against. Okay. All right, so here. This brings us to this idea of active and passive humoral response. Okay, humoral immunity. Remember, humoral has to do with antibody production. Okay? So here, when you have an active development of antibodies, basically, you are going to have, let me see how I can put this. Your cells are going to develop the antibody. Okay, so how is it you're going to have this humoral response? You can have it in two ways. Okay, your cells are going to be exposed to the virus, and you are going to develop your own antibodies. So that's active development of your antibodies. One way that you can get it is if you get exposed directly to the virus. Okay, so this is a natural way that your cells are going to develop the antibody against the virus. Okay. Another way is if you get exposed to a piece of the virus, but that piece of the virus does not make you sick. Okay, so this is really the principle of vaccination. Okay, in both of these cases, your B cells are producing the antibody, actively producing the antibody. One way is by directly fighting the beast. The other way is by fighting, I don't know, a little finger of the beast that got introduced into your system. Okay, so this is principle for vaccination, in which you supposedly do not get sick, but your cells are producing the antibodies. Okay. Another way that you can get antibodies in your system is by passively receiving antibodies. So there are also two ways of getting passive antibodies. When you think passive, it means that you get antibodies that were made by somebody else. Okay? Active antibody production, your cells make it. Passive antibody production, you got antibodies that somebody else made it. Okay? Who is somebody else? We also got two types. It could be your mom. It could be a transfusion of antibody through serum of somebody else. Okay? So in the first case here of natural passive humoral immunity is when you are in utero, right? In uterus, and you're connected to your mom via the umbilical cord and the placenta. Your mom is going to produce antibodies, mainly IgG, they're tiny ones. They're going to go through the placenta, through the umbilical cord, they're going to get to you, and they're going to give you immunity once you're out of the uterus. Okay? So usually you acquire this natural antibody transfer during the third trimester of pregnancy. Which makes sense, because if you're going to hang around in the uterus for 6 more months, you don't need to be fighting things that are outside of the uterus, right? So around third trimester, you get those IgGs from mom. So this is a natural way of getting antibodies. Another natural way of getting antibodies is in case of breastfeeding. So you have some types of antibodies like IgA that go through the breast milk and reach the baby's system and protect from pathogens. So it's also natural, and somebody else is producing the antibodies. Okay, so it is a passive type of acquirement of antibodies. All right. So here it has to do with mom. Okay, this other form here, it has to do in case you were really, really sick and your body did not have enough time to produce antibodies. And if you do not get those antibodies, you're going to die. In this case, you borrow antibodies. I mean, you just take antibodies from somebody else. It's not like you're borrowing and you're giving back later. Right? You take antibodies from somebody else that already produced it and survived the pathogen. So we have seen a lot of this during the pandemic, right? You know what I'm talking about? Yeah, antibody transfusion or serum treatment, right? In which somebody gets sick and you have to wait 10 days for your body to produce those antibodies, and those antibodies are not even good because it's a primary response, right? But if you get blood from somebody who survived COVID, you spin the blood down, get the serum, put the serum on your system. So this is a passive way of acquiring those antibodies, but it's not very natural. So we call that artificial. Okay? So get those antibodies in your system to neutralize the virus, and then you can survive. And why is it that the doctors didn't just want to sit around and wait for you to produce your own antibodies? In case of COVID, first round of COVID. It was deadly. So you're going to sit and wait until your body produces enough antibodies. The virus might kill you in 7 days, and then you're going to get antibody on day 10. Don't worry. Oops. But if you're dead at day 7, doesn't make sense. And your antibodies not even that good, right? So second exposure, now your antibodies are good, but then it's too late. Okay. So artificially acquired is when you get antibodies from serum of somebody else who produced it. And it could even be animals, right? You can guess, yeah. Yeah, that's IVIG, yeah, the serum transfusion, yeah. Okay. And it doesn't really have to be humans. You could have some other species that could produce antibodies. We're not going to get into that. That's too much of immunology. All right. So here's a little chart showing you how this distribution of memory of immunity will come out, right? So whole thing related to immunity, you know that you have this one branch that is innate, everybody has it responds every time the same way, right? And then here when you get to the adaptive immune system, this is when you're going to have natural or artificial, right? You're going to have natural that you get from mom, or you get natural if you get infected directly by the virus, right? Then you have artificial when you get antibodies from somebody else, or when you get immunized against a particular pathogen. Okay, so here is just from the book. And we're going to stop here. I'll pick up on the antibodies on Wednesday.